Okay, welcome to our first biochemistry session. This is going to be Chapter 1 of biochemistry for USMLE Prep, and this is going to be nucleic acid structure and organization. So the first thing we have to talk about anytime we discuss DNA, RNA, proteins, these sorts of things are the central dogmas of molecular biology, right? You've heard it once, you've heard it a million times, I'm going to make it 1,000,001. This is the fact that DNA makes RNA, which makes protein. Obviously, I'm using that term makes. There's a lot that goes into each of these processes that we're going to break down in later lessons. And then we also have DNA, which will reform itself. And we have names for all these processes, right? The name of this one right here, this big loopy arrow, we call this replication. Okay. The process of DNA going to RNA, we call this transcription. And the process of RNA being turned into sort of protein is called translation. Okay, so transcription and translation, these sound very close, very similar, and so you have to figure out a way of keeping these sort of straight in line. And the way I like to think about it is that DNA is transcribed into RNA, right? RNA and DNA have the same language. They're nucleotides, right? So scription, write, okay? This is like a scribe in the hospital, right? They take what the doctor says, right? Whatever language the doctor says, and puts it on a piece of paper. That is writing in the same language, right? Translation, think about it as well with language. If you're translating from something from one language to another, right? Say English to Spanish or vice versa. Here you're actually changing the language. So the language for RNA is going to be nucleotides and nucleotide sequences. The language for proteins are going to be amino acids, right? So whole separate languages. Okay. Now there's this one process called reverse transcription. There's more for completeness sake. I'm including it here. We won't be discussing this a whole lot until we get to things like virology and the microbiology section. So we've broken down the central dogma of molecular biology, and now we have to sort of go through the cell cycle, right? So at certain points in the cell's life cycle, it's going to be making RNA from the DNA. At some points, it's going to be making proteins and that sort of thing. At some point, it may even double itself. Okay, so we have names for these phases as a cell goes through. And a good place to start the cell cycle, I think, is with the M phase. Right? Call this M phase. Well, that's an extra M there. All right, M phase. What M stands for is mitosis, right? You can even think of the M stage as being meiosis. Either way, they just keep it nondescript as the M phase. Okay. After the M phase, during the M phase, you take one cell, you split it in two, and each cell is identical. They're identical daughter cells. Right. After the M phase comes the G1 phase. Okay. G does not stand for growth. Right. So growth is one of the major things which happens during the G1 phase. But G1 doesn't stand for growth 1, it actually stands for gap 1, GAP, gap 1. So the gap 1 phase, this is growth, and we're talking growing the cell size, the membrane, the cytoplasm, even organelles. We're producing proteins here, and some cells that don't divide will sort of stay in this state. And they actually have a separate sort of dormancy in terms of the cell cycle than the cell cycle arrest. Okay? So G0, or gap 0, is cell cycle arrest. And these are for tissues that will not be dividing. So things like nerve tissue, Most of your nerves of the central nervous system, they don't divide, they don't have a normal cell cycle, they are arrested where they are. Muscle cells are the same sort of thing. Okay, so after the G1 phase, we go into the S phase. S stands for synthesis. Right? So synthesis. What are we actually synthesizing here? Okay, what we're synthesizing is DNA. Okay, we are synthesizing new DNA, or we are replicating. Okay, so after we've completed the S phase, we now have sister chromatids. Right, we have the same chromosome which has been doubled and is now contained in one nucleus. And what we do after the S phase is continue our growth, right, in gap 2. Gap 2 is simply the stage between the synthesis phase and the mitosis phase. So gap 2 is in between the synthesis and the mitosis, and this is where the cell continues to grow and prepare for mitosis. Okay, now this is pretty much the complete cell life cycle, and we call everything from gap 1, from the beginning of this gap 1 to gap 2, we call that interphase. So interphase. Okay. And there are checkpoints along the way. There are checkpoints going from G1 to S. There are checkpoints going from S to G2. There are checkpoints going from G2 to M that we won't discuss at this point. But these just make sure that the cell is progressing correctly. Notice interphase is roughly 90 to 95% of the cell cycle, and the mitotic phase, right, is only about 5 to 10%. So the vast majority of the cell's life cycle is spent in interphase. Okay. So as an aside, there are certain drugs, as sort of a pharmacology note here. Certain drugs are targeted toward the different phases of the cell cycle. We think of drugs which will target the S phase, like methotrexate or hydroxyuracil or 5-fluorouracil. Okay, you see that uracil, we'll talk about that in a moment. There's metabolites, for DNA replication. Okay, so if you disrupt the usage of certain metabolites, you're going to stop cell synthesis or the DNA synthesis, and the cell will go into arrest or apoptosis. We also have other drugs like bleomycin, right, which will target the G2 phase. G2, again, we know is after the synthesis phase, so there's already DNA that's been replicated. So what bleomycin will do is actually break down the replicated DNA and keep it from progressing. We have drugs which will target the M phase, right, so mitosis. This is something we'll cover in genetics, but mitosis relies heavily upon the formation of microtubules, right? You have to form these microtubules to separate your sister chromatids and that sort of thing, and if you can't form those microtubules or you can't break down those microtubules, then it's really hard to complete M phase. So that's a big mechanism for a lot of these M phase drugs. And then you have other non-specific drugs and other chemotherapeutics that will not necessarily target one part of the cell cycle, but will trigger apoptosis in some way. Okay, so that's the big picture. We've talked about the central dogma of molecular biology. We've talked about the cell cycle and when we start dealing with DNA, when we deal with RNA, when we deal with protein. But now we're going to start getting into the nitty-gritty, the structures. Okay. Now for the USMLE, you're not going to have to draw out a nucleotide from memory, right? You're not going to have to remember where the oxygens are necessarily or anything like that. But you do have to have an idea of what goes into the structure of the nucleic acid. And the first thing is the sugar. The sugar is a pentose. Pentose meaning 5. And so we number these carbons 1, 2, 3, 4, 5. Now when we number carbons, there's a lot of carbons in the nucleic acid, and we have to be specific as to which carbons are which. So all the carbons from a nucleic acid have a prime. So this would be 1 prime, this would be 2 prime, this would be 3 prime, 4 prime, 5 prime. That's how we describe the carbons of a pentose. That's part of the sugar of nucleic acid. Now we know that nucleic acids can be ribonucleic acids, or they can be deoxyribonucleic acids. So what does that mean? Well, let's write this out. Or deoxy. Oh, that's supposed to be an O. O. Okay. So a pentose sugar can also be called a ribose, okay, once it's circularized, like we have here. And a ribose is just ribose. And we have deoxyribose. Deoxy meaning no or less oxygen. Okay, so let's discuss this. Okay, so we have our pentose sugar here, and there are some attachments that go onto these carbons, right? The one that we are going to worry about first is the 2'. Can have an OH. 3', have an OH. And then this 5' has an OH attached. That's what that little doodad up there, that little line up top, that is where an OH goes. So we have carbon 1, 2, 3, 4, and 5. What we're talking about when we talk about ribose versus deoxyribose is what is attached to this 2' carbon. Okay? So if we have an OH attached, as we did, that is ribose. Okay? OH attached. If, in fact, we have just a hydrogen, right, so we're missing the oxygen, or we're deoxy, right, call that deoxyribose, right? So the only thing which really separates a ribonucleic acid from a deoxyribonucleic acid, or RNA from DNA, is the presence of a hydroxyl group, or an OH group, on the second carbon. So that's our discussion on sugars. So now we're going to talk about the bases, right? So we know that we have different nucleic acids, right? So we know that there are many nucleic acids. There's not just one nucleic acid, but all nucleic acids have a ribose or deoxyribose sugar. So what gives a nucleic acid its identity? Well, the base is what gives the nucleic acid its identity. And we have essentially 5 different bases that we worry about when we talk about RNA or DNA. One is adenosine, one's guanine, one is thymine, uracil, and cytosine. So these are our 5 bases. These bases contain nitrogen. So they're sometimes called nitrogenous bases. And again, you're not going to have to draw out the base, but you do have to know a couple of things about that. The first is which bases are purines and which bases are pyrimidines. Because when we talk about the DNA ultrastructure, the double helix in a moment, you have to know which ones pair together. A purine doesn't go with purine, a purine goes with a pyrimidine, right? So essentially we have adenosine and guanine, right? These are the purines. Then we have thymine, uracil, and cytosine. These are the pyrimidines. So purines and pyrimidines, right? Well, what's the difference? Well, purines have a 2-ring structure. And again, you don't have to draw it out, but here's kind of what it looks like. We have 6 carbon there, and then we have 5. There's like a nitrogen. There's a couple nitrogens over here. You don't have to worry about where the nitrogens go, but adenosine and guanine, they are the purines, and they have a 2-ring structure. So the pyrimidines only have a 1-ring structure, and there's nitrogens in there, and there's certain attachments, but essentially the pyrimidines just have 1 ring. Well, how do you remember this? How do you remember that adenosine and guanine are purines, and thymine and uracil and cytosine are the pyrimidines? Well, there's a little saying. The first one says, pure as gold. And so we have purines are adenosine and guanine. Sometimes that's just enough to remember that the other are pyrimidines. Or we can have another saying, which is cut the pie. Right, so cut the pie. So cytosine, uracil, thymine are pyrimidines. So that is our discussion of the bases. The last part of a nucleic acid that we have to talk about is the phosphate. So we can have our ribose sugar attached to a base, and the base will always attach to the 1' carbon of the sugar, and we call this the sugar plus the base. We call this a nucleoside, right? So adenosine is a nucleoside, guanine is a nucleoside, right? Once we add a phosphate, that is when we have a nucleotide, right? So where does phosphate go? Well, let's put it on our 5th carbon here. So this is our 5' carbon. And instead of having an OH here, we attach a phosphate group. So once we have a phosphate, a sugar, and a base all connected together, this is a nucleotide. And we don't have to stop here. So we can have adenosine monophosphate if we have 1 phosphate, or we can have adenosine diphosphate, even adenosine triphosphate. And that's one you guys have all heard of. This is ATP, right? So adenosine triphosphate is a nucleotide. It also just happens to be the major energy currency of the body. So nucleoside, just the sugar plus the base, right? Nucleotide is going to be the sugar, base, plus the phosphate groups. So the next thing we're going to discuss, now that we've gotten down the structure of a nucleotide, are how these nucleotides are put together, right? Because one nucleotide does not DNA make, or one nucleotide does not RNA make, right? We have many nucleotides put together to make DNA and RNA, right? So the first bond we're going to talk about is the phosphodiester bond, right? So what does this mean? A phosphodiester. We have a phosphate connected to 2 esters, right? So remember that we have our ribose, we have our base, we have our 5th carbon, and we have a phosphate attached. This phosphate from this nucleotide will attack the 3' OH group from an adjacent nucleotide, right? So here's a nucleotide here, again, base, phosphate. So we have our 5' phosphate attacking the 3' OH group, right? It's a very important concept to understand, the phosphate attaching the 3' OH group, because we'll soon learn during DNA replication how DNA polymerase will attach these nucleotides together. Okay? So that's what is being depicted here in this photo, is a 5' phosphate attacking the 3' OH. We don't have the OH here. The phosphate, again, is always on the 5' end, and on the 3' end, and I have it depicted, is an OH group. So we have 5' phosphate, 3' OH. That's how you can identify the directionality of RNA or DNA. The next thing we have to discuss is how the 2 separate strands, right? So we have one strand here, we have another strand here. We know that DNA is a double helix. How the 2 strands of the helix interact with each other, right? And that's where we have sort of the ladders to our helix. Notice we have adenosine here, and we have a thymine. We have a purine with a pyrimidine, right? Again, pyrimidine with purine, okay? And there are these dotted lines in between. What are these dotted lines? These dotted lines are hydrogen bonds, okay? So we know that A, or adenosine, will pair with either thymine or uracil, depending on whether we're talking about DNA or RNA, right? DNA has thymine, RNA has uracil. Then we have guanine paired with cytosine. And just like the picture, we have 2 sets of dotted lines between the adenosine and its complement base, either thymine or uracil. And we have 3 dotted lines connecting the guanine with the cytosine. Well, this is the exact number of hydrogen bonds between the 2, right? So between adenosine and thymine, there are 2 hydrogen bonds. Between guanine and cytosine, there are 3 hydrogen bonds. Okay? So that's pretty much it for the nucleotide bonds. Again, we have a phosphodiester. Phosphodiester, they call it a backbone. Phosphodiester backbone, connecting all the nucleotides together. Then we have hydrogen bonds, which connect the two strands of DNA between each other. Structure and properties of DNA. When we think of classical DNA, we think of the Watson-Crick model, right? This is DNA that is a left-handed helix, right? And it's got roughly 10 turns, 10 nucleotides per turn, and that is bDNA. Okay, that is classically what we think of as bDNA. And when Watson-Crick discovered the structure of DNA, that's what they found was bDNA. Right? bDNA, this sort of thing, is not the only DNA out there. Right? We also have this stuff called aDNA. Right? aDNA is also a left-handed helix, but it's a really wide, highly coiled helix, right? So then we have zDNA, right? zDNA is a right-handed helix. It's a right-handed helix, right? So bDNA, stereotypical left-handed helix with about 10 nucleotides per turn. aDNA is the shorter, fatter, highly coiled DNA structure. zDNA is going to be a right-handed helix. Along with that, we have major grooves and minor grooves, right? So if we draw out sort of a DNA here, and I don't know if I'll be able to draw this well, but right, DNA is not really twisted around the central axis. It's actually sort of wrapped around. You can imagine, a phone cord being wrapped around a broomstick, right? When you have that, there's going to be major grooves and minor grooves, right? Major grooves having more space between adjacent parts of the DNA, and minor grooves having less space, right? And again, this is differences in expression, right? Proteins and leucine zippers and all that sort of thing can get into major grooves, whereas minor grooves are less prone to things from the environment getting in there and altering DNA structure. The last thing when we talk about structures and properties of DNA is the fact that DNA can be denatured. Right? What does this mean? Denaturation means that the DNA strands can be separated and then re-annealed. Right? So we have DNA sort of a ladder. Right? Given proper conditions, right? Certain pHs or heat, we can actually separate the DNA into two separate strands. Right? Which becomes very important. When we, again, talk about DNA replication, we have to separate the strands in order to copy it, in order to copy the DNA, but we have to also be able to reattach the strands together. Very last topic is DNA ultrastructure, right? If DNA was not condensed down into any form, it really wouldn't be able to fit into a nucleus, right? We have, meters of DNA for every cell in our body. It has to be really, really highly condensed in order to fit into a little tiny microscopic nucleus, right? Well, how do we do this? Well, we have these proteins called histones, right? Histones are rich in things like lysine and arginine, right? These are positively charged amino acids, right? Because DNA is negatively charged, right? So the positively charged histones will associate with negatively charged DNA, okay? The structure of a histone is what we call a heterooctamer, and I like to add of 4 dimers. So it's a heterooctamer of 4 dimers. Now what the heck does that mean? Heterooctamer means it's a protein which is made up of 8 different subunits, and those 8 different subunits are not the same. They're heterogeneous, not homogeneous, right? And how we get these 8, right, is we have 4 dimers, right? A dimer has 2 protein components, right? So 2 times 4, we get to an octamer, 8, okay? So you can imagine a circle, right, with 1, 2, 3, 4, and then we split each of those in half. So there's 2 1 subunits, to 2 subunits, to 3 subunits, and to 4 subunits. There we have an octamer, right? And those come together to make a histone. So now that we have our histones figured out, how do they interact with DNA? Well, we have our histone here, and the DNA will go and wrap around that histone, and then we have another histone, DNA wrap around that, right? Because the histones are positively charged, DNA is negatively charged, so the DNA will easily associate with it. And we sort of get this, what they call beads on a string appearance, right? And this is a nucleosome. So a nucleosome. But this isn't it. It's going to take a lot more than just wrapping the DNA once or twice or a few times around a histone in order to get the DNA condensed enough to fit into a nucleus, right? So what happens here? Well, this nucleosome will then start coiling around itself. In this coiling is on a fairly specific, fairly specific diameter. It's going to be about 30 nanometers wide. Right? So we call this 30 nanometer chromatin. And lastly, the 30 nanometer chromatin will be wrapped around and turned into a chromosome. So we know each cell in the human body has 46 chromosomes, right? 23 pairs of identical chromosomes, 23 pairs of chromosomes that we get from each parent, 23 from your mother, 23 from your father. Each chromosome is a coiled bundle of 30 nanometer chromatin, which is a coiled bundle of these nucleosomes, which is simply DNA wrapped around many times these histone molecules. That's a pretty complex condensation structure. So how do we get into that? How do we break into this structure to be able to express any genes? Well, here's where we discuss euchromatin versus heterochromatin. Euchromatin has an E, so I like to think of it as expressed. Heterochromatin has an H. I think of that as highly compact. Right, so a heterochromatin, highly compact. Euchromatin is expressed. So euchromatin, if you look under this underneath the microscope, you won't be able to see any chromosomes, right? The DNA is not nearly as closely associated with the histones here, right? And there are various ways the DNA can become dissociated from the histone. You can have deacetylation and these sorts of things where you will change the histone conformations so the DNA will become less well associated with it, and then it'll allow proteins to get into the DNA to allow it to be expressed, right? So that's euchromatin, this sort of loose chromatin that can be expressed as genetic material. And then you have heterochromatin, right, which is highly compact DNA, and you'll see heterochromatin whenever a cell is looking to undergo mitosis, right? You want to really condense down the genetic material as much as possible so that you can split the two sister chromatids apart. They go easily. You also see heterochromatin during cell death. So that's what concludes our discussion of DNA structure and organization. And I hope to see you for Chapter 2, where we'll discuss DNA replication and repair.